Good morning, dear friends. So, today our topic of discussion will be under the broad theme of developmental biology. Under this, we will discuss on the ultrastructure of sperm and ovum and also the process of spermatogenesis and oogenesis. At the very first, what is gametogenesis? Because sperm and ovum, as it is known to us, they are gametes. So, the creation of highly specialized sex cells through a process called meiosis. That is, we can understand gametogenesis is the process in which highly specialized sex cells, that are the sperms and ovums, are produced, and they are produced by the process of meiosis. Then gametogenesis is the formation of sperm and ovum. Spermatogenesis, which is the formation of sperms, that is male gametes, while oogenesis stands for the formation of female gametes, which is ovum. Thus, we can understand clearly from our previous definition that sperm and ovum are the specialized sex cells. In the process of gametogenesis, there is, that is, in spermatogenesis and oogenesis, three main phases. The very first phase is the period of multiplication. We have seen that in our from our previous knowledge, for any cell divisional process to be complete, the very first stage is multiplication. So, in the period of multiplication, what happens is that the primordial germ cells multiply by mitotic cell division. In this case, the primordial germ cells, or we can basically consider them as the precursor. As we know, for everything to begin, there is a initial stage or initial cell or any structure from which the process begins. So, here the primordial germ cells refer to the spermatogonium or oogonium cells from which the sperms and ovum developed. The period of growth. During this phase, both oogonia and spermatogonia, here oogonia and spermatogonia are the primordial germ cells from which the process starts. They grow in size. And the period of maturation. During the period of maturation, both the male and female gametes mature to objectify or achieve their target. In this diagram, on the left side, you can see the process of spermatogenesis, which is represented diagrammatically, and the oogenesis on the right side. So, let us enter into the details of this process. The male genital system. Before entering into spermatogenesis, we will take a brief outlook of the male genital system. This is necessary because spermatogenesis occurs in males. So, if we get a clear idea of the male genital system, we can have a better understanding of the process. You can see in the diagram several structures which are labeled. From the very top, if we consider the urinary bladder, seminal vesicles, prostate gland, the area of vas deferens, epididymis, testis, scrotum, penis, and urethra. This means all these organelles or organs have some specific function, which is why they are associated together to form the male genital system. Let us take an overview of the structures and their function. So, what is testis? Well, it is the male sex gland located in the scrotum. Here we can see that testis is a sex gland. Many a times we confuse ourselves with the term testis and penis. Many think that testis is the reproductive organ, or someone refers it to as the ejaculating organ. In that sense, we have to rectify that testis is not the ejaculating organ. The ejaculating organ is the penis in males. So, the testis, they produce sperms and androgens. Androgen is the necessary hormone which helps the process of spermatogenesis and the transport of sperms during the process of fertilization. We also have the interstitial Leydig cells which produce the sex hormone testosterone. The seminiferous tubule, which is also a structural unit of the testis, and it has the developmental phases of sperms in addition to Sertoli cells. Next we have is the epididymis. The function of epididymis is to store sperms. The vast difference which you have seen in the earlier figure, a tube-like structure helps in the transport of sperm from the scrotum to the prostate gland, while the seminal vesicle secretes fluid which is rich in fructose to semen. While discussing the structure of sperm, we will clearly understand the importance of seminal vesicle as why fructose is necessary. Keeping in consideration the point of fertilization, we know that sperm is passed from male or reproductive organ to the female body, that is from testis to vagina. In this process, the sperm has to travel a long distance to reach the fertilization point, that is where it will meet the ovum in the fallopian tube. In that phase, a sperm is highly specialized structure and it does not have any organelle to produce food, that is for energy. In this case, what happens, the fructose which is present in the semen, that is the milky viscous fluid which is also termed as semen containing sperm, provides the energy to the sperm so that they can use it and reach their target point. The prostate gland or ejaculatory gland which secretes milky alkaline fluid. Now you can see a term called sperm activation. What is that? The milky alkaline fluid helps in sperm activation and also it helps in neutralizing or balancing the pH of the female vagina during the process of fertilization so that the sperms do not die out. Last we have the Cowper's gland or the bulbourethral gland which contributes mucus to the semen. Mucus is very essential so that it can travel smoothly without getting struck or to reduce the frictional effect during the transportation of sperm to ovum. Here we can see the diagrammatic structure of testis where you can see all the nerve blood vessels and how they are interconnected. In this diagram, at the very top left side corner we have a diagram of the testis as a whole which has been magnified. You can see in the upper right side image that the spermatocyte is formed slowly from the first we have at the outer line the cells of Sertoli, the spermatogonium which gradually converts into spermatozoa, then spermatid and spermatocyte. Okay. So the Sertoli cells, what is the main function of Sertoli cell? We can see that at all the stages of differentiation, the spermatogenic cells which are in close contact with the Sertoli cell provide structural and metabolic support to the developing sperm cells. The concept of structural and metabolic support is very much essential because without structural support the sperms cannot develop and without metabolic support they will not be able to survive. A single Sertoli cell is able to extend from the basement membrane to the lumen of the seminiferous tubule. The Sertoli cells provide a number of functions during spermatogenesis and provides the cell nourishment, molecular signals. They also support them in developing gametes in the system. These are also some of the function which is maintained during the process of spermatogenesis, that is the environment between the blood testis barrier is always kept at a homeostatic level so that the sperms do not destroy. If you remember, in the diagram you have seen that the testis is always held in scrotum as sac-like structure which is detached from the body. The reason is that the scrotum keeps the testis at a temperature 2 to 3 degrees less than our body temperature so that sperms are not damaged. They secrete substance initiating meiosis. For any process to begin, there needs a precursor or a stigma or stimulation factor which is provided by the Sertoli cells to initiate the meiosis process to complete spermatogenesis. They support testicular fluid and help in secretion of androgen binding protein which is and also concentrates the testosterone so that the developing gametes face no difficulties. They also help in secretion of inhibin which is directed from pituitary gland for controlling the process of spermatogenesis. Now we will enter into our topic spermatogenesis. So as we already know the definition of spermatogenesis, that it is the process by which sperms develop in males. So we can see the process of development of spermatozoids from the male primordial germ cells and their differentiation into spermatozoa under the stimulation of anterior pituitary gonadotropic hormone. In the definition, we have two parts where in the first part we understand the process, and the second part how the process is initiated. Let's study the process of spermatogenesis. What happens in this process of spermatogenesis is that the primordial gram cells develop into spermatogonia. This starts or occurs after puberty. Now we have to keep it in consideration that spermatogenesis do not occur since birth, it occurs after puberty. Puberty refers to the sexual maturation of an individual, that is during the adolescence period, which we term as morely the teenage period. After that the process of spermatogenesis starts. In this case, what happens, the spermatogonia form the primary spermatocytes which undergo first meiotic division for 16 days. Then they divide into two secondary spermatocytes, that is by meiotic division. In this process the meiotic division is completed and each secondary spermatocyte has equal number of chromosomes, that is either 22 autosomes plus 1 X chromosome or 1 Y chromosome. Each of these divides again, that is the second meiotic division, thus forming 4 spermatocytes. So here we can see from one primordial germ cell or one spermatocytes we have 4 matured spermatocytes, and the process continues. The structure of sperm, as we can discuss, because we should know what is the structure or outcome of the product of spermatogenesis. So the sperm is divided into 3 structures. The first structure is head. The head has 2 important structures. The first is the acrosome, the second the nucleus. The function of acrosome. The acrosome is derived from Golgi apparatus and it contains hydrolytic enzymes. The hydrolytic enzymes helps in penetration of the zona pellucidar layer, which is the outermost covering of the ovum, while the nucleus contains the single set of chromosomes, or you can say the hereditary genes for transmission to the next generation. The middle section is nothing but it consists of numerous mitochondria. As we already know, mitochondria is the powerhouse of the cell, and we have seen in our previous understanding slides that fructose is supplied with the cells. So what happens, mitochondria intakes or generates ATP from the fructose and provides energy for the movement of the tail, and the tail, which is a flagellum, it helps in the providing direction and forward motion to the sperm so that it can reach the ovum. The female genital system, in this case if you see, the female genital system consists of ovary, that is the female sex gland, and produce ova. If we see this diagram very clearly, at the left side top corner we can see the female genital system. The female genital system, which has 2 tube-like structure forming a bent U-shaped structure, gives the fallopian tubes, which is followed by oviduct, and you can also see the central mass position where we have the vagina, cervix, uterus and the connective tissues, and on the ovary is situated neither inside or outside, it is connected via the fallopian tube to the mass visceral part. If we have a magnified view of the ovary at the below portion in left side, you can see that the process of oogenesis starts from the primary follicle where it's developed. You please try to follow the clockwise direction from the bottom part, where you can see that the follicle forms primary oocyte, where it develops into a mature follicle, and during the process of ovulation it is released as ovum, and then the corpus luteum is formed and degenerated. The ovum which is released during ovulation is collected by the fimbriae through a wave-like motion and passed inside the fallopian tube. The process of oogenesis, as you can see in the right side diagram, is very clear and apparent that by the process of mitosis, the oogonium forms the primary oocyte. The primary oocyte then undergoes first meiotic division, where one is bigger, and that is the secondary oocyte. One oocyte has the maximum amount of chromosome, while another is with a little chromosome amount, which is termed as first polar body. Considering the formation of polar body, a question might arise that why this is an unequal division. We will discuss this after the process is completed. Again, if we see in the secondary meiotic division phase, the secondary oocyte forms a polar body and the ovum or egg with maximum cell content. On the other side, we can see the first polar body also divides into two polar bodies. So, the outcome of oogenesis process is one matured egg with three polar bodies. The question might arise, why so? Obviously, the reason is that the egg, which is at the last part of the fallopian tube, is fertilized by the sperm. The sperm has no cytoplasmic content when it enters the ovum. Just the head portion enters the ovum, where the nucleus is released. It requires 7 to 8 days for the zygote to travel and reach the uterine wall, where it is implanted. To survive these 6 to 7 days, during which also the cleavage part occurs, the cell needs lots of energy, and this is stored in the cytoplasm. Now, imagine if the cytoplasmic content was divided equally into all the ovums without the formation of any polar body, all the 4 eggs or ovums would have very little cytoplasm to survive for 6 to 7 days and support the process of cleavage. Thus, to prevent the damage or spoilage of all the hereditary cells, or you can say the female sex cells, the female reproductive system has developed a unique and excellent system where one matured egg is formed and the rest 3 polar bodies are discarded. In this process, the implantation process, or you can say the objective of reproduction, is achieved successfully. These are some notes which I have given. You can take a screenshot of this for your exam purpose of view. In this part, nothing extra is discussed. Whatever I have discussed, it is written over here. That is the period of, and as usual, the oogenesis also, like spermatogenesis, what we have discussed earlier, the period of multiplication, growth and maturation is same. An important point which is to be considered here is that a baby girl who is born has about 2 million primary oocytes in her ovaries. It is in contrast to the fact that in case of males, the sperms develop after puberty, that is after they attain sexual maturity, whereas in case of women or female counterpart, primary oocytes, the primordial germ cells which give rise to ovum, is present after their birth, which accounts to a huge number of 2 million. But it keeps on decreasing. 2 million, you can estimate it as 20 lakhs. By the time of they have the age of 7 years, it is reduced to 3 lakh, and the rest are reabsorbed in our body. Only about 400 to 500 oocytes are released or will be available as viable oocytes when she attains the reproductive age. The penetration of sperm induces the secondary oocyte and the first polar body to complete meiosis II. This is a very important point to be remembered, that only when the sperm penetrates the secondary oocyte or the matured egg, the first polar body will give rise to 2 more polar bodies. In this, you can see the structure of an ovum which has been tried to fertilize by some sperms from the outside. The very first layer you have corona radiata, then the zona pellucida, the vitelline membrane, and the ooplasm, where it has its its nucleus. The main key points of difference between spermatogenesis and oogenesis are given in tabular form for your better understanding, and I hope with this we end our today's class, and I have been able to clear your doubts on spermatogenesis and oogenesis process. Thank you.